Hello and welcome to today's webinar. I'm Rachel Bromley and I'll be your host. We are so excited for today's webinar sponsored by Nacarta, and we have some wonderful speakers here for you. But first, let me have I have some housekeeping slides for you. So we'll just get through that real quickly. If you need closed captions or otherwise known as subtitles, you can click Live Transcript in your toolbar, select Show Subtitles, and if you need to adjust the size of the text, you can do that as well. That is an accessibility feature if you're having a hard time understanding the speakers or hearing. Disclaimer, TMA webinars are for educational purposes. We encourage you to make all decisions about your care in partnership with your healthcare team, and any disclosures the speakers may have are in their slides today. This webinar is being recorded and will be available on TMA's YouTube channel at the beginning of next month. Please put any webinar appropriate questions in the Q&A box. Any questions put in the chat box may be missed. If you pre-submitted a question, we have a colleague that is putting that in Q&A as we speak. And of course, please take the survey at the end of the webinar to let us know how we're doing. And please let us know where you are tuning in from in the chat. We love to see how far our community reaches. We have a new resource section. You can find more information about the speakers, helpful documents you can download, and links under the resources icon. If you don't see the resource icon, then click the three dots in a circle above the word more, and you'll be able to find it. Thank you to today's sponsor, Nacarta. We appreciate your support of the myositis community, and we're interested to learn alongside with you today. And so today's moderator is Jen McNary. She is a rare mom and a patient engagement professional with over 25 years of experience helping to infuse the patient voice into drug development. She is here today representing the patient advocacy team at Nacarta to introduce our fantastic speakers and to take your questions at the close of discussion. So welcome, Jen.
Thank you so much for having us today, Rachel, and thank you to the entire Myositis Association team, especially you, Rachel, Paula, and Linda, for your ongoing support and partnership. Today, we have Dr. Christopher Stein and Dr. Ortman, and I thank them for their willingness to share their knowledge and to continue to be present here for the community. And thank you to the community. We know that there are a lot of folks dialing in, a lot of folks that submitted questions. So thank you for engaging, asking those questions, and continuing to push us for more treatments and more options. Here today, we're going to learn a bit more about clinical trial participation, maybe a refresher for some or new information for many, but specifically Nkarta's NK cell therapy as a clinical trial option. As mentioned, I'm Jen McNary. I'm the CEO of Canary Advisors, and we partner with Nkarta's patient engagement group to support the advocacy team. Today, we'll take an in-depth look at the latest research on NK cells and myositis, and supported today by our wonderful speakers, Dr. Lisa Christopher Stein and Dr. Rob Ortman. Dr. Christopher Stein is a professor of medicine and neurology at Johns Hopkins University and the director of the Johns Hopkins Myositis Center, where she has become a world-renowned leader in inflammatory muscle diseases. She's a dedicated clinician, researcher, and educator, and she's been repeatedly named one of America's top doctors and recognized with awards for her pioneering contributions in compassionate, patient-centered care. We're also joined by Dr. Rob Ortman from Nkarta. He is the Vice President of Clinical Development. Dr. Ortman is a board-certified rheumatologist and immunologist whose career has spanned academic medicine, large pharmaceutical companies, and biotech. Dr. Ortman brings deep expertise in clinical trial strategy, regulatory affairs, and cross-functional drug development leadership. And we're going to start off with Dr. Christopher Stein. So I'm going to hand it over to you to share your presentation.
Thanks so much, Jen. And if someone could bring up the slides, we can start.
They are loading right now. Just a moment.
Well, while you load them, I will thank everybody and say good morning, good evening, good afternoon. I think I've seen at least one person from the UK. By my calculations, that has to be somewhere around 11:06 p.m. So, wow, thanks for your dedication for being here. Thanks so much for loading those slides for me, Richie. And so, my charge today is to first really just reintroduce for some of you or introduce for some of you the idea of clinical trials in myositis, and that really sets the stage for a lot of what we'll talk about today in the NK space. Next, please. These are my requisite disclosures, which does include Encarta. Next, please. So, what will we review today? Globally, I really want to remind you why we're all here. So, why do clinical trials matter in the first place? I want to remind you about understanding the role of clinical trials in advancing safe, effective treatments for myositis and why your participation is so powerful. I need to tell you that I wear a lot of hats. One of them, as you heard, is so I'm the director of and the founder of the Johns Hopkins Myositis Center, but I also play a role as the chair of the Institutional Review Board. That is the body at my institution that's responsible for making sure that trials are safe for patients. So I think a lot about trials as an investigator, but also in the regulatory space and making sure that each trial is designed for patients and often by patients because your input helps us design those trials as investigators. So, how they are designed, we'll learn again about the phases of clinical trials, what it means to be eligible, and what you might expect if you are a participant of a trial. And then the reason we're really here today is to talk about a new frontier. Cellular therapy has really revolutionized the way that we think about trials in myositis. I'll talk a little bit about where we were, where we are, and where we're going. I've spoken to many to tell you about my own journey as a rheumatologist. In the beginning of the journey for me, I was so excited to hear about biological therapy, translating immunology in the classroom when I was a med student to the clinic when I would be a physician. And some of that excitement was somewhat tempered when I fell in love with treating patients with myositis, but we really had older therapeutic options. While we've done well, we certainly can do a lot better. And I'll even put some of my own quotes on my slides. You'll see what I've said along the journey is that we are entering, finally, I strongly believe, a new therapeutic era. So I might have had to wait 25 years from the start, but I'm so excited that I was here for the journey, helping to tell a little bit of that story in being an investigator in some of these trials. I'm super excited to tell you that. So, in that vein, we will explore some of the exciting science of NK therapy, what CAR therapy means, and a lot of that will be under Dr. Ortman's purview today. And this idea of a potential immune reset, it's kind of like a reboot of the hard drive of our autoimmune computer. All right, let's go forward. Hopefully. So I don't think I have to remind this audience, but just in case this is somewhat rare to you or you're very new to the to myositis, having been a newly diagnosed patient, we have dermatomyositis. I talk about polymyositis in quotes because I honestly believe it's an older term. Having said that, a lot of trials still include that subset. To me, that's often rolled into some overlaps and may or may not be misdiagnosed as immune-mediated necrotizing myopathy, which you see next on this slide, the antisynthetase syndrome, usually without a rash, or inclusion body myositis. And some of you, I'm sure, will ask about IBM, inclusion body myositis, how NK therapy is related. For better or for worse, most of this topic or talk today does focus more on non-IBM. However, I do think there's always lessons that we can learn for IBM related trials. So remember that I'll talk about IIM or idiopathic inflammatory myopathy. That's the broad term that really is myositis. Myositis by itself really just means inflammation of the muscles. But when we talk about autoimmune myositis, that larger term IIM is used in trials. And as you well know, many of you are probably more of an expert of this than me, having lived this disease, but this is still considered rare. It's a rare autoimmune disease subgroup in which the immune system can attack the body's own muscle, In the case of dermatomyositis, or sometimes the antisynthetase syndrome, the skin, lungs, and other tissues. And I showed to you on the right the key features of each of those subtypes. And so in MyoCon 2025, I was tasked to talk about clinical trials. And I think the exact quote from me was that for years, almost every major biologic tested for myositis has failed. Now, for the first time, I think we're starting to see the path forward. All right, next, please. So we know that current treatments leave many searching for better alternatives. Without clinical trials, we as physicians must rely on older, less specific drugs. Clinical trials are really the only way where we can test new medicines safely. And for the myositis community, the trials represent the primary mechanism for me for hope and improved care. Next, please. And as I told you, I play two hats. I have two hats. I play two roles. Clinical trials should be and are carefully designed around patient safety. While we all want efficacy, we all want a good answer at the end, the overarching goal of any clinical research study is to use science to improve human well-being. Participation should always be voluntary. I talk a lot about shared decision making. I try to be honest about trials for my patients and when they may or may not be eligible. When they are eligible, I think it's an important conversation about whether it makes sense for them, and we'll see why in a moment. There's revocable consent. You can always leave a study at any time for any reason. You are not bound to a trial, especially if you worsen during the trial. Certainly, it is always helpful for patients to continue throughout the duration of the trial so that we can get enough power in the trial, enough people present to make sure that we can answer the question posed by the trial. It's thoughtfully designed, right? Every step is engineered to try to keep participants safe throughout, and there's several checks and balances in clinical trials. Next, please. So your lived experience carries as much weight as clinical data. So, as doctors, we get excited about data. We get a little excited about things, and we want to make sure that we get so excited that we don't ask the patient. In my own experience doing patient reported outcome research, it was you that taught me how much pain is involved in this disease. And so we want to make sure that that is a surrogate endpoint in the trials that we study. We don't want to study the experience that we understand. We want to understand and learn about the experience that you tell us about. So it can be fatigue, pain, physical function, skin symptoms, etc. So trials need to measure what you as patients feel and experience, not just a lab test, not just a muscle strength test. Your voices in the trial design actually help make research genuinely patient-centered. Next, please. Oops. So, clinical trials should be science in service of patients. As I told you, these are designed to test whether a new treatment is safe, but also, of course, effective. There are they are the only scientifically rigorous way that we as investigators, as scientists, as physicians can determine whether something truly works. We do open label or our own little mini trials of look at our own experience as physicians in the clinic. But truly, they need to be designed in certain ways where we often look at them against a placebo, an inert substance, to see whether or not what we think we're seeing is really true. They're designed to improve care. Every trial asks a specific scientific question to advance how we treat and prevent illness. There are built-in safety protections. What are they? Well, I told you there are independent safety boards. There's ethics committees like the IRB, and there's the FDA in sponsored trials that monitors all of these trials continuously. I told you that your participation is voluntary and informed consent. You should receive full information about the risks, the benefits that should not be overstated, and the alternatives before you ever agree to participate. Next, please. So every new treatment must climb the rigorous staircase of safety, as we say. So this is phase 1 through phase 4. And the next slide is a bit more helpful, I think, as we can look at each of those stepwise ways to get to phase 4 from phase 1. Next, please. So the four phases of clinical trials start in phase one. Now, these numbers on the slide, by the way, are a little bit more skewed for the general population and cardiovascular diseases. If I could ever see 1,000 patients in a phase three trial for myositis, I would fall over. That would suggest actually that myositis was more common than it is. So in rare trials, we'll skew back these numbers. But I think you can see in general the iteration going from a few to a few hundred to a few thousands to post-marketing. So phase one has just a few patients, a few people involved. The focus in phase one is really safety. It's not like we can't look for some efficacy or a signal, and indeed we like to do that, but the focus there is safety. Is this a safe substance to give to participants? It's often given to healthy individuals, individuals without the condition. So we're evaluating safety, tolerability, and determining often the right dosage. Phase two is more people in those trials, and that focus is efficacy. The goal there is to look at further assessment for safety as well as efficacy and to determine the dosing range. So we've already sort of figured out, we think, our dosage, but phase two can help us look at multiple dosages, for example. Phase three is more patients. In myositis, I'm excited if a phase three has over 100 patients or participants, or 200, 250 is excellent. The focus there is confirming sort of what we're seeing from phase two. The goal here, confirm effectiveness, monitor the side effects, and compare to standard treatments. And then finally, phase four, we're not out of the woods, right? We're excited when we finally get to phase three, a drug gets approved, but we need post-approval monitoring. Every once in a while, a signal comes out only when more patients take this drug when prescribed clinically rather than in a trial. So it's our duty to monitor for long-term effects, identify rare side effects that might not have been seen until more people have been exposed, and ensure ongoing benefit risk balance where obviously the balance is towards benefits. I need to remind you that many, actually most, drugs that enter phase one may never ever reach approval, and it's frustrating. So this is so exciting when we have phase three results in myositis. Next, please. So without trials, we cannot move forward. So I told you, you know this, myositis is rare. We have historically relied on very old treatments. They're often borrowed from other diseases. In fact, other than steroids, really none of these drugs, even like methotrexate, azathioprine, you will maybe know these by their trade names, and many of you were started on them or may still be on them. Some of them work great, and sometimes that's all we need. But many times, especially in a specialty center where I practice, we need greater efficacy from something else. None of these were specifically tested or approved per se for myositis. They get grandfathered in, or they're approved based on other diseases, for example, but none of these were specifically trialed for myositis. Other major biologics, rituximab, tocilizumab, others either failed or were never even either failed in trials or were studied or wanted to be studied and never even made it to the trial space because the studies were terminated before they could launch. So there are over 12 phase 2 and 3 myositis studies underway in 2025 alone. Myositis is having its moment, right? I have been seeing my colleagues in psoriatic arthritis and other rheumatic diseases like rheumatoid arthritis have multiple drugs on label for years. And I've been waving a white flag, like, please, please look at us. And for some reason, and finally, during the pandemic, it seemed like people finally listened, concentrated on myositis, and we're in a good space. FDA-approved therapy specifically for myositis, still only one, and this is the Proderm trial that Octagam branded IVIG 10% for dermatomyositis was approved. It's a start, and I think it got people excited to say, wait, maybe we can get a drug to the finish line. If one is good, more is better, and people became interested. And who were people? Big pharma, small pharma, biotech, and so we're in a new therapeutic era. New therapies are on the cusp of FDA approval, hopefully in 2026. We have brepocitinib, which is in registration now with the FDA, and efgartigimod, which I'll mention briefly again, which is in trial now, and hopefully the results will be soon. Phase 2 was positive, which I'll mention momentarily. Looking for phase 3 results, and hopefully that may be one as well. And why we're here to talk about what we're here to talk about today is the idea of the first NK CAR trial, which is now enrolling myositis patients among other diseases. And you'll hear much more from Dr. Ortman. So trials give us hope, and patients who participate make this progress possible. We know you take a chance to be in a trial. We know that you're the pioneers that get something from A to B, and eventually, hopefully, it benefits more people. I am struck just personally by my own research efforts. I do natural history studies where we look at patients' journey in myositis. And I can tell you that virtually every patient who agrees to participate in research says, while I hope this will help me, even if it does not, I hope it benefits someone else. I think that is the most generous spirit, and I hear it time and time again. But of course, I hope when we enter you in a trial, it's going to help you. Next, please. Am I eligible? What are the general criteria? Again, not to not talk about inclusion body myositis. Inclusion body myositis is its separate entity. Happy to talk about that a little bit more. IBM has been frustrating for sure, and usually anybody with certain characteristics, as long as there are not comorbidities that prevent you from being in a trial. Comorbidities would be other diseases like brittle diabetes, something that is severe diabetes, heart disease, something where we feel it wouldn't be safe for you to participate. Most trials to date in inclusion body myositis included everyone, and the placebo group is justifiable because to date, other than exercise, there is very little that we have found to be effective. That may be changing, and we may have certain inclusion criteria for IBM that might include very specific cutoffs for how severe the disease is for trial entry. More on that, and that's probably a different subject. But for today, I'll speak about non-IBM eligibility. So, a confirmed diagnosis of myositis. That is done by confirmation of clinical criteria, classification criteria that is generally put forward by the bodies of people like the American College of Rheumatology or the European Rheumatology Associations, EULAR. Active disease. We can't enroll you if it's not active. Why would we do that? Would be unethical, and we want to be able to see progress. So ongoing muscle weakness and or skin involvement, depending on which disease subtype. This is a little frustrating because to date, most trials for dermatomyositis past phase 2 have necessitated skin and muscle to be active together. Many of you may be listening saying, well, I am a patient with dermatomyositis who does not have muscle involvement, or my muscle involvement is controlled, but my skin is not. We, myself, and others are trying to get the FDA and others to understand, and they are listening, that perhaps decoupling those, meaning that patients who have only skin disease may be eligible for trials in the future, and I'm excited about that. Muscle weakness documented by testing, and MMT is a manual muscle test, and that score is generally a cutoff of either 136 or 142 out of 150. I realize that means nothing to you as a patient, probably, but I can kind of tell you that 150 means that we're scoring usually 8 of your muscles, both sides, including your neck flexors and upper and lower body. We score that out of a 10. Sometimes you'll hear me, if you're in my clinic, talk about 5 out of 5. There's also a 10 out of 10 score for trials, which essentially is full strength. If you have 142, in other words, 8 points off that total score, you're considered usually weak enough to enter a trial. Some trials, however, require you to be weaker at 136. Prior treatment with at least one immunosuppressant, which might include steroids. In some trials, it's a steroid plus something else, and that's really for ethical purposes. We usually do not enter you in a trial at entry, meaning that's the first thing you ever try. To be honest with you, I think that paradigm is of interest in my own experience. Earlier treatment sometimes leads to better outcomes, but for now, unless it must be done systematically, where we unfortunately have to fail sometimes a few earlier drugs to be able to allow a trial. We are pushing that timeline forward a little bit more, not making people fail one drug after the next to get to hopefully something that would be more effective, or a clinical trial when other things are not. You have to have stable background medications. Why? We want to make sure it's not the adjustment of those medications that are leading to the outcome of the trial. So that's called a confounder, and we want to limit confounders. Rituximab use is generally not allowed in almost any trial and must be held for up to 6 months. Sometimes we'll check your B cells, which is a type of white blood cell that rituximab specifically targets. And if it's that those B cells have come back up to regular levels, normal levels, some trials allow you to participate. IVIG is generally held for 3 months, although some trials are allowing only a 1 month hold. That is the number one reason that I have had some difficulty in recruiting, is that patients may be incompletely controlled on their IVIG, but don't want to stop it for fear that they'll lose control in entering a trial. And that's a real concern. I understand that. And so we can talk about that maybe in the Q&A and ways to make that better. So the 1 month hold has really helped people feel more comfortable with entering a trial. What can you expect? Well, you usually can continue your steroids, sometimes, usually methotrexate, mycophenolate, or azathioprine, not usually combinations, and not usually a biologic therapy like something like rituximab or other biologic therapies. Some trials include a placebo period anywhere from 6 to 12 months, and that's hard. It's hard to say that you're on a placebo. Generally, in these trials, you're not on a true placebo, meaning you're not getting anything. You're getting your background therapy, but you're just not getting the active drug. You're getting an inert substance as a comparator. Autoantibodies are the response against certain proteins that you make. Many of you may know your own autoantibody. I'm a big proponent of understanding autoantibodies. How does the body see these, your own proteins as abnormal and make a response against them? They are now thought to be pathogenic. They're actually causing the disease. And so knowing that status sometimes is required, especially if the way the drug works is to lower that antibody. And then finally, skin scoring, something called a CDASI, a clinical score of severity and extent of skin disease, may be used in dermatomyositis trials. Next, please. I'll go quickly through this because a lot of this is going to be covered by the next talk. Just wanted to show you that today's trials aim to block inflammation. They can block this by blocking interferon, by an easier delivery system of IVIG, by targeting complement. Some of these trials did not continue, but were started and we learned from them. Efgartigimod and nipocalimab are, for example, blocking the recycling of antibodies, mimicking at least some of the effect of IVIG. And today our subject is, of course, NK CAR, and I'll go forward with the next slide there, please. New wave of targeted therapies. What you just saw on the last slide is the same thing here. So again, mechanisms are diffuse. We are looking at different ways to block different parts of your immune system. I will not belabor these, you are not an immunologist, but just to tell you that we're looking at every part of the immune system. And I say that we've gone from a cannon, right, shooting out this target that just we hope that when we block your entire immune system or large parts of it, that we get the bad part, but also you take down the good part. This is why infection can go up. This is why we get off targets to the target of your own disease. And that is frustrating for us. To me, this is the same paradigm of chemotherapy, where we use just broad chemotherapy in so many cancers. And now our oncology colleagues really are showing us the way to have targeted, very targeted therapy, trying to eliminate more of those off-target effects, making us just look at tumor rather than the entire your entire body. Similarly, we're trying to look just at that part of the immune system and not the entire immune system. Next, please. So the current challenge we have is continuous treatment without a true immune reset, right? So complete depletion within the target tissue is difficult. We're trying to target the organ that is most affected, skin or muscle or sometimes lung, but you're burdened with the need for continuous ongoing treatment. B cell depletion, for example, that one part of the immune system, that white blood cell, it's vastly changed the treatment landscape. It can lead to disease remission in some patients, but not all. And that's probably because we're not getting as deep as we need to go and where we need to go and as continuously as we need. Go on, please. So I will probably save a lot of this, knowing that we're getting close to my timeline. But just to say that this is the trial that we'll talk about next. I'll leave this in Dr. Ortman's capable hands. And what is exciting, I think he will speak about, is that most of the CAR T therapy, which is akin to this therapy, is done in an inpatient setting. This therapy, as of April, I believe, the FDA has allowed outpatient administration now, which is reducing your monitoring time from a day of 24 hours to just 2 hours. This is revolutionary for CAR T therapy, for CAR therapy rather, where we cannot usually, we can't do this in an outpatient setting. And even though in theory, CAR therapy may be absolutely revolutionary for some patients, executing that has been complicated. We need to borrow oncology space. We need to ask our oncology partners to be generous with that space. And we need new systems and a little less fear as rheumatologists to navigate side effects and different systems in being able to deliver this therapy that is unfamiliar to us. Next, please. So the goal is not just control. I've talked about this immune reset, and I'm going to skip this slide because I know it's going to be covered. And then so finally, we are at a turning point in myositis research. B-cell depletion has changed our landscape. New targeted therapies are arriving. We talked about those. And this shifting paradigm of CAR NK therapy represents this movement from lifelong immunosuppression toward either one-time immune reset, hopefully, or at least one-time, less often reset. And I think we're on the verge of hopefully something transformative. So I've said, others have said we're not declaring victory yet, but for the first time, we can see a path forward. Next, please. So your voice shapes the future of myositis research. Measure what matters to you. For me, a trial must capture what you feel and experience. I don't care what your enzymes do if it doesn't matter to you. Fatigue, pain, skin symptoms, quality of life, for me, are real, measurable. And actually, the FDA now requires a patient-reported outcome. So it is organizations like the Myositis Association that advocates for your voice to be heard. You are the expert of your own disease, and your experience informs trial design, better endpoints, and ultimately better treatments. Next, please. So what can you do? You're in the most exciting era of myositis research to me in history. In 2025 alone, as I've said, we've had a dozen or more trials that were either underway, some were stopped prematurely, some went on. We learned a lot, but 2 new therapies are likely on the verge, hopefully, of FDA approval. And the first CAR NK cell therapy trial for myositis is now enrolling. Talk to your doctor, visit clinicaltrials.gov. Not my favorite site for navigating trials, but does tell you what might be available. Join a registry, share your story, and support patient advocacy groups like the one that's hosting this webinar today. Next, please. So I'd like to thank you for your attention. I know I went over by just a few minutes, and I will wait for the Q&A after Dr. Ortman's presentation. Thank you. Thanks for the clapping. Thanks for the love, and thanks for your attention tonight. I look forward to the discussion. Thanks so much.
Just a moment. Pulling Dr. Ortman's slides right up.
And you didn't go over Dr. Christopher Stein. You were absolutely perfect. So fantastic. Thank you so much for that presentation. And as mentioned, Dr. Ortman is going to continue the discussion in just a moment.
There we go. So thank you, Dr. Christopher Stein, for actually teasing and leading into this discussion. I was kind of bummed that you didn't go over those slides, because those are the exciting ones for me in drug development. So for everybody else here, I'm Rob Ortman, the Vice President of Clinical Development at Nkarta, and want to thank the MFA for allowing us to be a resource for them and for us to share what we're doing in our development program, and for the number of you folks here today that are interested in what Dr. Christopher Stein has said, and for what I'm going to be explaining about why we're excited. So now we move from the academic to the publicly traded company. These are our forward-looking statements, an eye chart. So I showed it. Let's go to the next one. More. We've disclosed everything we need. So as Dr. Christopher Stein alluded to, we are an NK company, Nkarta. And NK cells are natural killer cells. And what we have done here is taken these cells and engineered them to be effective treatment, or hopefully be effective treatments in autoimmune diseases. And we believe that NK cells, which are your first line of defense, are a better approach to treating disease than the cell types that really kick this off. A little bit of history, those were T cells. So NK cells and T cells are part of your immune response. And what initially was done a number of years ago is that researchers engineered the receptor of T cells, and that's what's chimeric antigen receptor, to basically recognize antigens, proteins on the surface of abnormal cells. And we in immunology were fortunate that they focused on B cell lymphomas, because as Dr. Christopher Stein alluded to, B cells are very important in a number of our autoimmune diseases, including myositis. So what we believe we have is we've engineered a better approach to CAR therapy that will be better for patients. One of the aspects that we think we will be able to do is be able to give this in an outpatient setting, not requiring overnight stays or extended stays. And we tell our physicians, we hope this to be similar to rituximab or Remicade, except instead of pulling it out of a refrigerator, it comes from a freezer. And the last one is that how we're able to do this is we don't have to engineer your cells to be able to recognize the protein we're looking at. We're doing that in the back room. So we're doing that all at our facility so that the cells are available to be given in the outpatient clinic. So next slide. So again, I think I've already touched on this. It's on-demand availability. You don't have to wait for the engineering. We've clearly shown that we can do this in the outpatient world. As Dr. Christopher Stein again mentioned, the FDA has agreed to allow us to do that. The third checkpoint there is really important when we compare CAR NK to CAR T cells. When patients receive CAR T cells, it's very important for those cells to expand in the body rapidly so that they can do their work. And unfortunately, you may end up with some very untoward symptoms. One is cytokine release syndrome. Cytokines are messengers that help fight inflammation, but when there's too many of them, it's like having a bad fever or the flu. And ICANS is when you have those symptoms predominantly in your nervous system. So we think that with the CAR NK cells, you don't have that higher risk. And then finally, the potential for redosing. Like a number of CAR T and NK cell companies, there was a first foray into oncology, B-cell lymphomas, and we were able to see in our oncology program that we were able to successfully redose patients. We are not the first CAR NK company in the autoimmune space, but we are the first one in the myositis space. A company, from China has done some excellent work in refractory SLE and showed that they were able to get these patients into low disease activity or remission in 2/3 of the patients at 12 months. And again, had no evidence of these toxicity syndromes. And again, I think what we're looking at is trying to achieve something similar in patients with myositis. Next slide. So again, we're talking a lot about B cells. B cells are the cells that make the autoantibodies that Dr. Christopher Stein was talking about that end up attacking your muscle cells. Back in the day, when I was training, everything was polymyositis or dermatomyositis. In the intervening time, what we have found is a number of these diseases, including dermatomyositis, polymyositis could be really broken apart into autoantibody specific diseases that actually cause disease. So it's very important to get rid of those B cells that are making the autoantibodies that cause the disease. And so what we're trying to do here is set up something called immune reset. So the chart on the left, the time graph, is what happens when you are treated in this fashion. It's a little bit different than what Dr. Christopher Stein has shown for most traditional drugs, in that we start with stopping most all of the agents you're on in order to harvest the T cells from patients and engineer them while the patient's still in the hospital, and then reinfuse them. One of the reasons that drugs are stopped and then something called lymphodepletion is given is to try and create a space where the CAR cells can, in this case, expand, as you see by that peak, and do their job. And their job is basically to kill B cells. There are a number of antigens on B cells. The one that's most commonly targeted is CD19. That's the ones that were involved in lymphomas. And so in that middle part there, these CAR cells are very busy getting into the tissue and destroying all those bad B cells that make the autoantibodies. One of the reasons we don't think rituximab is very effective is because it doesn't get very well into the tissues. We're able to see some B cells depletion in the blood, but as our surgeons and pathologists say, tissue is the issue. We need to get the drug, in this case the NK cell, to the affected tissues where we can wipe out the bad players. And then one of the markers we have for doing adequate B cell depletion is the emergence of what we call naive B cells. So you could look at the old autoimmune B cells as being rogue pirate cells that are there to wreak havoc, and we get rid of them. But part of the immune response, we need B cells, and then we get emergence of the naive B cells. So it's kind of like the end of the year, out with the old, in with the new. And there are ways that we can check for that. And the nice thing that we're seeing is in the CAR studies, most patients are getting this naive cell reset. So in particular, we have a study called NTRUST-2, a number of, it's a basket study. This is where we look at a number of different diseases in one group. As Dr. Christopher Stein mentioned, myositis, although everybody here is very familiar with it, overall it's relatively rare in the population and even in rheumatology. So we try to study patients who have rare disease, myositis. Some of you may know people with scleroderma or systemic sclerosis and a certain vasculitis in a basket study. And so we're looking to enroll those patients. Would be happy to chat further about that with you in the Q&A session. But I just want to give people an idea of what CAR NK cell studies can look like, what we're trying to do, and how we think it can be beneficial to you. So I think that's all we have now. And again, if anybody's interested in getting in touch with us, there are two email addresses there for you to reach out to. And that's all I have. And I'll turn it back and we can enter, I think, the Q&A session.
Absolutely. Thank you. And hopefully Dr. Christopher Stein is still here. Good. So I'm going to go through some of the questions that are pre-submitted and also entered in the Q&A. And as a reminder, please feel free to enter your questions. Even if we don't get to them, you can reach out to the patient advocacy email address or to the Myositis team, association team, and we'd be happy to get those answers to you. So one of the pre-submitted questions is about NK cells. And the question is, are these cells currently existing in the body, or is this something like a serum that's injected in the body? And I don't know, Dr. Lisa Christopher Stein, do you want to try to answer that first, and then we can head it over to Dr. Ortman?
Yeah, I mean, someone said killer sounds scary. I know, right? But like you
Yeah.
want it to kill things sometimes. You actually want killers in there if they're going to kill something that's bad for you. So you want a natural killer when it's going to kill the right thing. You just don't want it to kill the wrong thing. So, that's part of what sort of there's the innate immune system. They are like, I guess, like an early responder. They kill a lot of things. They'll kill a virus infected cell. They can kill cancer. So they are sort of like I'm trying to think, how do I say this as a not like a not an immunology audience? They're kind of like signaling molecule that shapes your immune response. So there's sort of a training of the immune system, and they don't require that same training that a T cell requires, for example. So yes, they're there to protect you. And when you engineer them to kill or to protect you from your own immune response, they can act in the same way they would be for a foreign invader. I don't know, Rob, if I've done a good enough job of sort of saying it from,
I think you did. Um, you know, they're natural killer cells, and they're kind of the liaison between, your I don't know what to call it, polymorphonuclear, your neutrophils, which when you get a staph infection, get sent there to try and kill the bacteria. These cells are, in the normal people and everybody generally are looking at trying to get rid of cells that have abnormal, antigens on the surface that are common when there's a viral infection or cancer. And so they're in surveillance. And so what we're doing is taking advantage of the fact that they kill cells in a specific way. And what we do is we grow them up back in the lab, and we engineer them to be specific. That's the CAR part for antigen on B cells called CD19.
So I know we already answered this, but I did mark to answer this. The K stands for killer. That's scary sounding. So Rob, I don't know if you have anything to say about, why the K, why killer cells?
I think partly because when they were first studied, as I mentioned, they had a very impressive ability to destroy infected cells that we really didn't see with other cells. And as I mentioned, the natural part is that they don't have to be trained like T cells and B cells. So they're kind of the commandos.
So somebody's asking, how can we get this treatment? And as a reminder, this is currently in clinical trials. And so the answer to that is you can talk to your physician to see if this might be a good fit for you, but also on this screen here, you can see the clinicaltrials.gov. Again, this is a place that nobody likes to go, but that's where clinical trials can be listed. Or if you'd like me to give you a link, you can email the patient advocacy at Ncarta.com email address there, and we'd be happy to point you in the right direction for where you can go to learn more about this study. Thank you, Linda, in the chat. So here's another question, and we'll start again with Dr. Christopher Stein. How should patients evaluate emerging therapies like NK cell treatment when reading about them online? What's the best way to evaluate it?
Yeah. So first of all, I want to be clear that the reason I say that clinicaltrials.gov is not that I don't want to go there. I just think it's a difficult system to navigate. Yeah. I mean, I definitely think it's helpful. I just wish it were a little more navigable. And I've had a lot of thoughts about how to redesign the way we think about getting patients clinical trial info.
It's odd because that whole site was set up to be patient-centric to learn what's going on with trials. So
it's not even doctor-centric. I mean, I have like 100 years of education and I can't find these trials. It's a little frustrating, but it does provide info. You're just not always sure if they're actually enrolling, what is enrolling. So I just want to be clear that yes, it's reputable. It's just difficult to navigate. That's been for years. So after that, I've forgotten the question. So you're saying, how does someone evaluate a trial or how?
Yeah.
So I think a bit of it. So I think, you always want to have, trust but verify and have some healthy skepticism. I mean, I think that as an investigator in a trial, and I'm required, for example, when I present things to you, to disclose my conflicts of interest. I'm technically conflicted with a lot of a lot of organizations because I am paid to do consulting or research or speak sometimes. Although I'm not a paid speaker per se, I am a paid consultant. And so you want to make sure that your doctor or the person that is enrolling you in a trial has your interest at heart. I think it's clear that I want to be very transparent. I am, not. I am careful to make sure that you enter a trial based on your own volition, that you want to do it, that it makes sense for you, and that I advertise it to you. Everybody who is part of a clinical trial, usually people who are part of a clinical trial are compensated. I want to make sure that the compensation is not an inducement for you to enter a trial. Outside of that, the trial itself, sometimes even though you would want to enter, there's reasons that you should not or could not enter, like you don't meet the entry criteria. I think when you're looking at a trial, the most trusted person, hopefully, should be your treating physician to be able to go over this with you. If they are not enrolling in a trial, experts in the disease, these organizations like the Myositis Association, which I think does a wonderful job trying to break it down in a non-conflicted fashion where they can tell you each trial and what they're designed to do, who can enroll, and to speak to your physician. I think that's really my best advice to you is ask your physician if you think that you should join. Quite honestly, I think it's our job to advocate for the trials, but I have found that even in as an expert, even in an academic practice, sometimes we might make decisions for you. I find my own bias for that and trying not to do that, where I assume maybe a patient wouldn't want to enter a trial because it would be difficult, like travel. Sometimes travel costs are covered. Often travel costs are covered. Maybe difficult if you have children at home. Maybe difficult if you have elder care, or maybe just be difficult for you because you're physically unable to do a lot of the travel that's required. So certain things like this, I think that we might have some biases, but I think it's both the clinician and the patient's obligation or duty to speak to each other, to express interest, and for me as someone who does a lot of clinical trial work, to make sure that I appropriately ask patients. I also try to present all trials that we have available that would be the person would be amenable. And if there's more than one, have the patient in shared decision making with me make that decision. If there is only one, I may state, look, there's only this one is the one that you currently qualify for because disease X is covered, or you're this weak, or you have the right skin scores, those kind of things. Um, yeah, I mean, I think you have to be careful. Should you use things like AI and other instances? I think you need to be careful, but again, maybe trust but verify, and make sure that I have my own patients type out things or print out things from the web, bring them to me and ask if sometimes they educate me and they're the first people to tell me something's available. I try to be the person that tells them, but you all are super smart and sometimes bring me things and I learn. So I think it's reasonable to educate yourself and then ask your physician.
Absolutely. This question's for Dr. Ortman, I think. So thinking about NK cells, are these therapies using a patient's own cells or donor cells?
Good question. These are called allogeneic, which means they come from somebody else, versus autologous, which is when those are the cells that are engineered from your own blood. So they're, again, like I say, two different approaches. We've taken the allogeneic approach where we get cells from healthy volunteers, engineer them so that they two things that they do is they respond to this CD19, and that's what activates them to be the commando. And then I also say they bring along their own lunch. We've engineered a cytokine, a growth factor, so that the cell's able to stimulate itself. So this is allogeneic, from somebody else. And we also, I think it's important, we try to match for some of these blood types, we'll call it, to make sure that it's not rejected as well.
And I think we have time for another question, one more, because I'm going to wrap two questions into one because they both also have the CD19 reference. So somebody wants to know, how do we know that CD19 cells are the apparent cells in dermatomyositis, but also how do you make NK cells to specifically target CD19? And I don't know, Rob, if you want to take that question first.
Sure. Again, how do we know that it's CD19 cells? Well, basically, there's been a lot of research in B cell biology over the years to understand what they're doing. And the primary purpose, I will say, of B cells is to make antibodies to protect you. And a problem with autoimmune diseases is that these B cells go rogue and make autoantibodies that can harm you. And so we just, from our understanding of these proteins on B cells, as they go through their growth stages, CD19 happens to be one that's present for a long part of their growth cycle, especially up to that point where they start making these autoantibodies. So it's been a very attractive target from that perspective.
If I could just jump in, just because we might have made a leap, and I think that maybe for people, and I'll tell you, I loved immunology, but it's one of the most complex subjects in medical school. I think I still always need an immunology refresher, just for patients. And I always really try to educate patients, and I believe every patient can understand, to some extent, your own body. I believe you should know the CD19 is it's a marker on B cells, and B cells have this large, lineage from baby B cells to mature B cells, the whole thing. And so CD19, CD20, these are markers on a particular B cell. We can see them in your biopsy of your muscle, for example. So we know they're in the target tissue. We know they produce antibodies, and those antibodies have been shown recently in very elegant experiments to actually almost certainly be responsible for the disease. We used to think they were innocent bystanders. We're not 100% sure what B cells did, how they do the damage they do. I think there's more off targets to B cells than we understand, but they are the primary driver. CD19 is the marker of that B cell that's going to make that antibody, that self-protein response that is the damaging part of the disease. I just wanted to jump in to say like that, just in case you're wondering, weren't we talking, what is a B cell and what is CD19? CD19 is a marker. There's a lot of cellular differentiation, CD, that stands for these different markers on all types of cells in the B cell pathway.
Thank you so much. And I think we have a brilliant group of listeners today. Those questions were fantastic. So we are at time. We always want to go as long as we can, but we are at time. So we're going to hand things back to Rachel for the close. Thank you both so much.
Thank you.
Thank you.
Yes, thank you so much for being here. That was so informative. We really appreciate everything you do for our audience today and beyond. I've just got a few slides to share. And again, we would like to thank our sponsor, Encarta, for sponsoring today's webinar. We appreciate your support. We don't want you to miss these upcoming events. We've got Myositis Research Insights webinar featuring Dr. Panal Fernandez, and that'll be Tuesday, June 9th. And then on Friday, June 12th, we have a meet and greet leadership series, the MyoCon edition. So all things conference related. If you're going, if you're not going, if you're on the fence about it, this is a place to come together and just meet up with your fellow people. So it'll be fun. That one's a fun activity. And of course, these slides aren't loading, but I can tell you that June 17th is a Valor Study update webinar that we have. And then on June 18th, we will have Dr. Rob Hollowell, a pulmonology edition of Ask the Expert. So we're very excited about that, and didn't get to see the graphics I worked so hard on. Anyway, we also have support and affinity groups. We want to let you know this Saturday we have our myositis support group by diagnosis. So each diagnosis has their own breakout room, plus the care partners meet in their own room. And so that is a great support group meeting to go to. And then MyoCon's going to be in St. Louis. You can see the arch behind me, and early bird registration closes July 1st. And one last appeal to take the survey so we can improve our programming. And again, thank you and have a wonderful rest of your day. Bye, everyone.